Thank you very much for the invitation. It's been so lovely. I got to see a little of countryside yesterday, which was very nice, and now to see this university is amazing. It would have been nice, I think, if I had already visited before, because I don't know. I have no idea what kind of your challenges are. And so part of what I wanted to do today was to get some ideas from you. Rich asked me if I would talk a little bit about how I deal with teaching in the large classes in particular. And I don't have any pictures of them, except I have a few videos that I'm planning on showing you. So to get a glimpse of some of the challenges that I've worked with and struggled with, and then maybe some of the solutions that I share today might be helpful. One of the things that I use a lot in that enormous classroom is the electronics to help me get input from students. And so that was what I had here. We use a program called Top Hat. There are others that are available that are free. For me, I use it as an incentive to get students to answer questions. And I found that unless I offered points associated with this assessment, then they didn't do it. So if they purchase it. But for today, you guys are going to use a demo course. So I was asked if I might do a brief presentation at the beginning on what is active learning, what are we talking about. And I work with National Academies of Sciences in the United States. Created these summer institutes in biology teaching. And so I've been working with those for years, helping, we go around and we have workshops of just about this exact number of faculty that come for a week, and we work with them to help transform their courses. So I'm going to try to do a very brief version of that just to give you a flavor of what some of the things are that might be possibilities. And I'll blame it on them. So some of my goals for this session were that I would show you some evidence about the effectiveness of more student-centered or active learning teaching strategies, and then talk a bit about using backward design to align some outcomes that you might have for your classes, and hopefully at the end give you a brief amount of time to start thinking about how you might modify some of the things that you're already doing in the classroom that would help sort of rise to some of these challenges. And we'll talk a lot about deliberate practice and what that means. So I'll start this by saying not everyone's in the sciences, and certainly you guys are not in the United States, but in the United States, our real goal for transforming the way science is taught at the college level is to improve not only student learning, but also retention. Retention is one of our biggest issues. So this is just a chart showing how many STEM majors we start with in the United States, and then we tend to lose about 40% of them. And this is a concern because we really, our president has recently come out with a report saying we need 1 million new STEM graduates, and if we're losing a lot of them along the way in various stages of the pipeline, this is a big issue for us. One of the things that they've started to look at is when these students are leaving, and discovered that a lot of times the students that we have that are abandoning a science major are leaving in the first few years. And at universities in the United States, the culprit for that has really been very large lecture courses that we teach for our introductory students, chemistry, biology, and physics. And so there have been studies over the past 20 or 30 years that have looked at what some of the problems might be in those courses, and lots of reforms that faculty have started to do to change those courses. And so Scott Freeman, who works at the University of Washington, did an analysis of some 255 different studies and published this in the Proceedings of the National Academy of Sciences in 2014, and found that students in the traditional lecture, when it was compared to students that were taking these more student-centered active courses, were about 1.5 times more likely to fail the course, which is a big issue for us. And so as a result, we've started developing more active learning classroom structures like this, where you could rearrange the students and have them work more intensely rather than just sitting and listening to a lecture. And in these kinds of classrooms, not only were they less likely to fail, but they made about, on average, about 1/2 letter grade higher. Now, you might think, well, but maybe in these classes, the course material is just delivered in a different way, and it's true. Students have more opportunities to earn points on activities that they might do in class rather than on end-of-course tests that they might be taking. But if you look individually at the data, for example, those classrooms are called scale-up classrooms, and they were originally developed in physics. So this is data from the force concept inventory in physics, which is a kind of end-of-course test that students are all expected to take. And it's showing in the sort of light blue how students did in terms of gains over the course of the semester. They were given it at the beginning of the semester and then at the end of the semester, and then they looked to see what the normalized gains were. And you can see for these scale-up classrooms, these are mostly at North Carolina State University, where Bob Buechner has sort of pioneered this use of this kind of classroom technology, but also at the University of Central Florida and other campuses. You can see that in general, students in red in the scale-up classrooms are making greater gains over the course of the semester on those kinds of end-of-course assessments. But what's interesting, and I think I kind of take this to heart, because we worry that we're preventing the best students, right? In our sort of push to get all of the students to be retained in the sciences and move upward, we also want to remain concerned that our very best students are also getting the kind of challenge that they need. So in these different charts, B, M, and T were students that were in the bottom, middle, and top of the course, and it's looking at the gains. And what's really interesting, so this is using the MIT electricity and magnetism test, that students in the top tier, they aren't making very many gains in a traditional course. They've come in with a lot of knowledge, they don't have a lot of room. But in this kind of more interactive scale-up classroom, where students work together on more challenging problems, they're able to make even greater gains. So this was definitely something that we were interested in biology, right, to try to figure out how we might be able to incorporate some of these gains that students were seeing. And what we ended up thinking about were the fact that these classrooms allow students in exchanging information and in particular kind of conceptualizing what does knowledge mean. And so I want to do a brief, quick exercise with you guys about the sort of just showcase the value of these conceptual frameworks. And this is from How People Learn, was written by Chase and Simon in 1973. And so this was one of the experiments that they described about the value of learning. So it's called the chessboard challenge. And in the challenge, they showed participants this picture of a chessboard with the chess pieces on it. And they gave them a few minutes, and I'll give you guys like 15 seconds here to look at it. And they asked them to memorize where the chess pieces sat. Okay, time's up. And then they gave them a blank board, and they said, please place as many of the original chess pieces as you can onto this board. So they gave them that. There were 22 in that last picture. How many are you guys getting? None. 6. Good. Okay, so what they did this is they actually selected 3 different kinds of participants. They chose chess masters, they chose Class A players, and then beginners like me who just watch other people play chess. So what would your hypotheses be about how well these individuals did on this task? Beginners the best? What do you know? No, right? Chess masters. So that's what they found. So this is the number of pieces that they recalled correctly. They had 25 total pieces. We only had 22. And then they gave them different trial numbers. And you can see that the chess masters there I have black words, in red are our chess masters. They started out getting about 15 of the chess pieces on their first try. They could improve it by the second or third try, but then after that, they had pretty much maximized. So they were able to recall almost immediately where all these chess pieces were. And then the Class A's did a little bit better. And the beginners, it was just trial and error. So you don't feel bad if you were the same as them. They only got 4 or 5 pieces on the first try. And then repeated trials, they could finally maybe get close to where the masters and the Class A players were. So this sort of brings up this issue of why. How can people be taught to recall information better? And what's the difference between what we see experts doing and what we see a total beginner, like the students we have in our classes, doing? And so they gave them also this chess challenge here. And then these are the results that they got. So anybody here who plays chess know what the problem is? Yeah.
Too many pieces.
Too many pieces, and and they're random, right? It's not a game that someone's playing, right? So a chess master who goes in can look at it and say, oh, I understand exactly what they're playing. But here, where the pieces are just randomly assorted on the board, it becomes, in a way, the way students who are new to this come into your classroom. They don't understand why you're telling them these things in this order or what they really mean. It may make total sense to you, but it's sort of like you have to go backwards in time and imagine what it's like to learn something if you have no context and to kind of put this information in. And that's this idea of this contextualizing what it is that students are learning. So this is a lovely picture. It sort of diagrams what we're talking about here. And our experts on the right has both the concepts and also the connections that are meaningful between those concepts, and they can access information by relating them to each other. Whereas our novice has limited understanding or whole missing concepts. They may have some concepts that they have, but they may not have connections between them. And they might have a lot of wrong concepts that they're constantly sort of going back and forth and trying to see how they work. So I thought I would ask you guys a little bit of our quick top hat question to see, okay, if you were trying to get someone to go from being a rank beginner to a Class A player to a novice, what kinds of things, what kind of tasks might be most effective for those students to look at? So I think if you look at Top Hat, you can open this up and it will let you rank these. If you don't want to rank them, you can talk them through to the person next to you and find out, what do you think? Where would these go? So I have this up here. This is what it looks like from the instructor's perspective. And then later, I think when we do some short answers, you can see that it will also scroll by and you can see what other students in the class are asking in terms of questions that they might have for a discussion. So it's kind of nice because it allows you to actually be engaged in what's going on. And then for me as an instructor, I can use it even in a class of 300 people, and I can tell when everyone's done. And when we should sort of maybe start looking at some of the responses. And so it's got that sort of percentile here of the percent that have finished, and then it will also generate reports. And so I can click on reports and it can quickly show me that most of, like 25%, we have interacting with peers to make explanations, and lower down sort of using simulations. And then finally at the bottom, I'll scroll down, it's probably listening to peers, interacting with peers, etc., down from the top. So I brought this up here so we can.
Yeah, I think you can't read them, but the first set it starts with interacting and goes on.
Each bar is a
yes, kind of where they are, in what order they're in. I'm going to close this. We'll go back over to here. So the reason I brought this one up is that very often when you think about there's this long-standing myth about how much people recall of sitting and listening to a lecture, how much people recall of watching a television show or a video. And so there's been work over the years to try to figure out what should I be doing to maximize the time that I'm having with students. And so there's a lovely article that came out that was a synthesis of some of the research that has been done called Multimodal Learning Through Media, what the research says from Cisco Systems. And it was really looking at how well do students learn when they're using different kinds of media, and multimodal, what about when you combine two forms of media together? So they're watching an animation with a narration that comes over. Maybe they're looking at text with illustrations. And what they're interested in, and this is really important because you think about the chess challenge, you have some basic skills, like I can just recall where the pieces were, possibly greater, I understand what they are, to more higher order skills. I want students to analyze and evaluate and create and judge and problem solve. All the things that we're really hoping they would get out of a college experience. And so this is sort of showing how well do students make gains in either the basic skills or in these higher order skills using non-interactive multimodal learning. So that includes not just text alone, but text with illustrations, or not just animations, but animations where you're also listening as well. And you can see that students make about 21% increase gains in basic kinds of skills in recalling and understanding things. And about the same really for higher order skills, so where they have to analyze and evaluate and judge things. Where some of the other skills come in, and I put some of those on the list for you guys to choose from, is what about when you've got a little bit more interaction going on, where you're discussing it with a peer, or you're having a real-world lab experience where you're trying to put things together. Well, what's interesting, and I think this is something that people are often very challenged with, is that for basic skills, you just don't really see a significant gain in terms of where the students are. Where you see the kinds of gains are what Rich was talking about with those higher order skills that he was hoping his students would have. So the question for every instructor when they come into a classroom is really, what is it that I want students to accomplish? Where are they? And what the best modes might be for them to do that. So if you want students to be able to remember things and sort of basic skills, it may be great to have animations and text with illustration. You don't really need to have classroom discussions and interactions with their peers to get that kind of basic knowledge. But certainly, if you want to try to work towards some of those more higher order skills, you really do need to put students in a situation where they have to grapple with a misunderstanding and try to put those concepts together. So I put this up here because I know this is an active learning session, and I don't want you to come away from it thinking, oh, I think everything should just be peers interacting with each other, because I don't think that's true at all. What the challenge really is how do you get all of this learning to happen in your classroom? How do you make spaces for all of these different kinds of learning to happen? I will put a push, though, in addition to the active learning, in general, coming from trying to retain students in the classes. So it seems like it's, as I said earlier, it's helping with our failure rates and also the grades that students are getting in classes. But in the US, right, we're supposed to be making these million new STEM graduates because employers want to hire them. And so there was a lovely survey that was done. It's called Falling Short, College Learning and Career Success. It was a survey that was conducted of 400 employees of the Hart Research Association, American Association of Colleges and Universities, last fall of 2014. And they did a variety of different questions that they asked these future hiring people that we're hoping our students will go to. And they said that the vast majority of them thought that it's important for all colleges to ensure that students are prepared with skills needed to complete a significant applied learning project. And unfortunately, only 14% of employers think that the college students are prepared. So not only are we challenged with graduating more students, they really want them to have the kinds of interactions where they can do these maybe more applied learning in situational contexts. And I'm sure Sarah is going to talk a lot more about that as well. So what does that mean? It means that maybe we need to start thinking of our classrooms less as lecture halls and more like a coaching experience. So within coaching, so this is this picture of this is US college-bound high school students who are practicing football, right? So they have a coach here, and he's not sitting down and lecturing to them for the entire semester about the game and what the rules of the game are and what the plays are. They certainly do that. They have playbooks, and that has its space. But in addition, he's also hoping that they'll go through and practice some of the skills that he wants them to do through sort of deliberate practice where he's there observing them, he can give them feedback, and sort of help them work toward developing all of the skills that they need. So what I wanted to do is show you guys what that looks like in the classroom setting. And this is a video that I made that I took, but is now available on iBiology, which is a series that has been created trying to get more opportunities out there for faculty to look and see what's happening in the classroom.
People often ask, what does it mean to teach well? What are we talking about when we say to teach well? I think this comes back to the question of instructor-centered versus student-centered teaching. Student-centered teaching, for me, is an attitude more than anything else about how to go about preparing to teach a class and how to actually teach it. And when I used to lecture traditionally, I spent a lot of time preparing for class. A lot of work went into it, but what I was thinking about always was, what am I going to do first in the first 5 minutes, and then what am I going to do after that, and then what slide am I going to show, and so on. And the student-centered approach, you're turning it around and asking, what are the students going to be doing during the first 5 minutes? What are they going to be doing during the next 5 minutes? What's going to happen after I give them this problem? So that the focus is on what the students are doing in the class and not on the professor.
So, to start understanding sex differentiation, we're going to watch one more video, and then you're going to work on an assignment of learning in groups. I want you to start, there's some reading at the top, and then I want you to work on number 1, which is on the front. Just ignore number 2 for now. You're going to want to make sure number 1 is in good shape first. I want you to work on it alone to start with. So, learn to work in groups. I want you to discuss your figures and come to some sort of agreement about what they should look like. Group number 19. Yes, what answer did you have? Right, okay, so this group is thinking that this individual developed with some of the female secondary sex characteristics. So let's vote on that. Let's vote folders. How many groups think that this is going to develop as a male? So this individual would certainly develop a male reproductive system, although there are other genes involved in this besides the SRY.
So it goes on after this. I'll stop it there, but you guys are welcome to go and watch the full later. It goes on to show how she deals with the other questions that come up. So they do both sort of pages of a workshop. I'm hoping that you could see from that when I think of active learning in the classroom, it just means providing that opportunity for students to practice what it is that you want them to be doing eventually. So maybe it's to test their understanding of what's going on. So in the case of the clicker questions, you could ask a quick understanding question. But in many cases, you want also for them to explain why they're confused. So the question is really a means to sort of identify where the missing pieces are and where the incorrect and confused ideas are. So it gets them to sort of open up. And you can see in that classroom that she also has them working in permanent groups of students so that they're more comfortable talking to the people around them and sharing where those confusions are, and are more likely to raise their folder and have one of the members of their group maybe explain to the whole class why they chose a specific answer versus another answer. And they can get help that way. So I guess before we move on, I kind of wanted to open this up to you guys and ask a little bit about what concerns you have. So you've seen I've given you like sort of rationale for why this might be effective, and then shown you what it might look like in a classroom. I have no idea for you guys, what does that mean in terms of what concerns you might have? So we'll see how this one works. We'll open this up. And the question is really kind of what concerns do you have about using this in your own classrooms? And it will show up as anonymous, so you can take a minute to think about this. And I will say that getting to know someone who does this and contacting them is extremely helpful, even if it's someone who's doing it here on your own campus, who's just starting to do it. And the other thing I'm going to push for is to start small and just see what happens. Because you don't want to just throw everything out. You want to just try something and see what goes on in that situation. The best thing is that no matter what you do, you will learn a lot more about where your students are confused. They may not be comfortable with it, and you may not be comfortable with it either, because it's a little disturbing. But once you've realized that they don't get what you're talking about, it becomes pointless to move on, right? You think, why would I move on when they're all completely confused about this? They haven't gotten this basic thing. I don't see the point in moving on now. And so it kind of convinces you. You do have to do some serious thought about what the most important things are for you to consider spending time on. And I don't think that that's ever a bad thing, to think about your whole class and to try to prioritize what some of the most important things are that they're doing. I know that Sarah, luckily, is going to talk about some of these several groups in the same class, so I can worry about dealing with some of those later. These are fantastic answers to questions. So this is what I had planned to do, and I think that it will probably work. I wanted to show some examples of some smaller things that you can do in the classroom, and to talk in particular about how do we find the time to make this work in the classroom, and how do we do that. So in general, the suggestion that's made goes back to that chart about how much students make gains in learning when they're doing an interactive kind of thing with peers, or if they're doing something that's maybe just some multimodal. They might be looking at text with animations or text with figures. And that is to take the stuff that can be learned best out of class and shift it out of class, and to hold students somehow accountable for it. There's lots of technology that's available in terms of online quizzing and tutorials that you can make do. Will students cheat at them? Yes. Yes, they will. But you've made the effort, right? And you can try to make these things with multiple questions. There's all kinds of arms race kinds of things that can happen to ensure that you're rising to the challenge of students. But I also think that once they realize that there is something meaningful and interesting going on during class, it will and maybe inspire them to want to get some of this stuff done outside of class so that they can actually do something more meaningful in class. Usually the out-of-class stuff would be the things that they can learn very well in that setting, the fact-based, knowledge-based, recall kinds of things, and further practice, additional problems that you've given them. For example, I'll think back to the math derivation. Physics has been doing this scale-up kinds of classroom for decades, and there are many different textbooks available that have tons of simulations, animations, questions that allow you to push a lot of that content onto the online format and to use class time then where the instructor can really work one-on-one with students. And that's what you're going to try to put in class. It's going to be deliberate practice of whatever the most difficult concepts are for students and involve some critical thinking skills. And that involves mainly questions. It's the same kinds of questions that you would give students on a test, but maybe modified into a format where they could do them in class. So, when we think about this, I'll sort of ask this one. We don't necessarily have to do a top hat poll. Read this scenario, this description. Imagine this is you. And you're trying to do some classroom teaching. What do we think's wrong with this scenario? Right, so some of the major issues, it's just, if she can't even remember it, why are we doing this? Why are we taking class time to sort of do this? Which is exactly right. And so, no matter what you did, you have to think about the content for content's sake, right? Are we just covering stuff because we're supposed to, or is there ever going to be a time where we think, is this important? So here's another. Let's imagine, what's wrong with this instructor here? Okay, so you got it right, though. The idea is that sometimes, and this is true, people think if the students are actually doing something, then I've fixed the problem, right? They're actively doing something. And in this case, right, it's supposed to be about what they're learning, and it doesn't matter if they're completely active, they're not learning anything. So when you guys are planning to do something new for your course, let's say you were going to add something to your course based on thinking, okay, I want some more student-centered, maybe some higher order thinking. Where do you start? Where do you usually start?
With what you want to learn at the end.
Okay. He's anybody else? Be honest. Outcomes. Oh, you were too good. Come on. Really? Really? Because what I used to do was I would think, okay, it's photosynthesis today. We're going to do photosynthesis. And I'm going to think of stuff I want them to do in class. And I never really thought, what is it that somebody really needs to know about what this is? So you're way better than I ever was. So I had to kind of unlearn all of these things. So most teachers would basically just start with, well, this is what I want students to do in the class. And what they were going to do to make that happen. And right. Instead, I needed to start thinking about your perspective, which is at the end of the course, maybe even 5 or 10 years after the course. Is it important for them to know the clades? Or is it important for them to understand where these electrons are moving through this chain? I don't know. And if I can't convince myself that it's important, I think I need to abandon it. I think I need to move on. And instead, really focus on things, maybe a question or a concept that I know they're struggling with, but that I'm not willing to get rid of because it's too important. It's actually critical to understanding whatever that concept is. So this is this idea of backward design, which is we're going to start at the end with the outcome. By the end of the class, what do they need to know? What is it that I think is really critical for them? And then what kind of evidence am I going to be able to gather that students can do this? So traditionally, that's our tests. But we might think, well, yeah, but I don't want to wait to the very end of the semester and discover they didn't know anything about this, because then I can't go back and fix it. It's too late at that point. So instead, I'm going to start to think about the activities in class that will help me get to that outcome. And hopefully, I'll provide some opportunity where I can see them practicing in front of me, and I'll be able to adjust what I'm doing and what they're doing so that I can address where they're confused and what kinds of problems they're having. So we make ourselves start to think backwardly, starting with the outcome. And I'll show you guys an example. This is, I teach this class for introductory students who are taking biology to fulfill their requirement for graduation. They do not want to be scientists. They do not want to take the class. They do not want to see me. But they are forced to in order to graduate. So a very hostile kind of group of students. And you think to yourself, they don't need to know that. They don't need to know that. They do not need to know that. So it makes me take a very, very kind of critical look at what it is I'm wasting their time on. And for me, I really want them to understand, what do scientists do? How do they know what they know? And in particular, in this class, there are things like enzymes, which are really critically important to how the body functions, how living organisms function, and a lot of the medicines they take are sort of based on understanding enzymes and how they can be blocked. So that was really important for me. So then I started getting a little more specific, thinking, well, I would like them, if they looked at a table or graph of data, I want them to be able to interpret it. And then I want them to use an enzyme as an example to relate what a change in the gene for an enzyme might result in the organism. Like, how would it affect the organism? So those were my outcomes. And then the assessment that I used at the end was really related to that. I wanted them to use a data or data in a table of frequencies of certain genotypes in the population to explain if the presence of a specific allele predisposes someone to developing a disease. So you think, allele, I don't know, I've forgotten all these terms, I have no idea what she's talking about. I'll show you an example in a second. And then that's what I decided to have them do as an activity. Let's actually have them grapple with some data in a table and then come to a conclusion based on this table. So I'll give you the example. So here's the test items that I actually asked them to do. I said, moderate alcohol consumption, 1 or 2 drinks a day, has been identified as a risk factor in developing breast cancer, something they might come across in the media. It's known that individuals differ in their ability to metabolize ethanol due to genetic differences in the enzymes that catalyze the reactions shown above, and I give them the reactions. Some researchers have hypothesized that the increased risk of cancer might be due to high levels of something called acetaldehyde, or just aldehyde, a compound recognized as a probable carcinogen. Which of the following enzyme variants would lead to higher acetaldehyde levels? I'm thinking, I don't even know what she's talking about. And then describe the data that would convince you that a specific combination of alleles in genes encoding these two enzymes would result in an increased risk of breast cancer in women who consume alcohol. So this was what I wanted them to be able to do. If they were reading an article about the risk of increase for cancer, that they would understand that. Now, what I'm going to show you is if you did a search on Google, like my students will on their phone, for alcohol metabolism or enzymes that digest alcohol, they would come up with this. This is one of the first hits, Lord of the Drinks. Now, this is a blog, right? This is where students get their scientific information. And it is entitled, Why Indians Are Alcoholics and Asians Can't Drink. And it has this derogatory picture, and it's full of all kinds of real issues and problems. But if they were to Google, this is the kind of information they would come up with, right, from their search. And if you look in a little closer, it has some kind of scientific words, exactly like the scientific words that I was using in some of those assessments. When you drink alcoholic drinks, the ethanol is metabolized into something called acetaldehyde. This is being done by an enzyme in your body called alcohol dehydrogenase. So the more ADH, alcohol dehydrogenase, your body produces, the faster the process. Apparently, a lot of Native Americans miss out on this enzyme, according to analysis on chromosome 4 and 11, and that increases the risk of becoming alcoholic. Unlike the American Indians, the Chinese and Japanese produce a lot of ADH. 85% of their population produces unusual high activities of this enzyme. So my thought was, I'm going to read this, and of course they're going to believe it. They're going to think, first of all, there's no data given here. There's no information. So I thought, well, what if they did search and they actually found one of the articles that these people are referring to? Would they be discerning? Would they be able to look at the data in that article and distinguish whether or not it had any truth in it? So this gets us back to what it is, like a major role that I want them. But within this, there is stuff about the genetics of this that you have to be able to understand. And there's also stuff about metabolism, which is a basic part of biology, that were fundamental issues that I wanted students to get. So I show them this, and we read a case about a couple of students who get messed around with some alcohol, and some of them get alcohol poisoning, and another one gets a very flushed face reaction, kind of feels sick and doesn't want to drink. And they then have to consider, why is it that someone might have a better tolerance for alcohol, and what that might mean in terms of the developing alcoholism later in life. So this is a portion of the lecture that I give to them. So you can see the slides are in white. So first of all, I have them draw out on a piece of paper what happens when you consume alcohol in your body. I discovered that my students thought that the alcohol that you ingest in a drink comes out in your breath, right? Because they've been taking breathalyzers. So like, it comes in my body, it moves around, and then it goes out in breath. And I was like, okay, all right. And then I talk about, where else might it go? Great, that's awesome. Then they say urine. I'm like, all right. Okay, so it's all in there. Yep, they're totally satisfied with that. It comes out of my breath or my urine, and it's gone out of the body. And so I said, well, let's consider this. How come you can get a beer belly then? You drink a lot of alcohol, and suddenly you get fat in your beer, in your gut. So why is that? I never thought of that. Okay, so then I start talking about the role of enzymes in the process, and I say alcohol is food, it has calories. So it must be digested just like the other food in your body. And so I show them this graph, which shows that they're very aware of, because they've had alcohol training in the United States to make sure they don't drink and drive and things like that. And it shows here blood alcohol concentration over time. And it's color-coded by how many they would have had. And so I said, what's going on here? This is very interesting. Tell me what's happening. So in a group, they discuss it. So what do you guys see happening in this graph as you're interpreting it? What kinds of things are going on? Does the alcohol stay in your body a long time? Pretty long. I mean, depending on how much you drink. If you had this, it's going to be 4 hours later, you still got pretty high alcohol level. This is 0.05. If you blew that in your breathalyzer, they would be concerned. But it's clearly going away, right? It's disappearing. So how is that process happening? Well, then I say, well, there's an enzyme that's responsible for that. That's what they were talking about in the article. Generally, enzymes, here's our little E for enzyme, take some kind of substrate, S, and through a process, chemical reactions, converts it into a product. And so in that graph, they were basically showing you the disappearance of the substrate in that reaction. So this happens, thankfully, in our bodies, thanks to liver and intestinal cells that make an enzyme, which is called alcohol dehydrogenase. And I describe it as like a little Pac-Man. And its mouth binds to the substrate, in this case, the ethanol or alcohol, and converts it into this product, which is called acetaldehyde. So we've discussed this. We've talked a little bit about enzymes and how they work. And so we can sort of think about the process of breaking down alcohol a little more fully. So alcohol is broken down into acetaldehyde by this enzyme. And we can call this oxidized or broken down. But it turns out there's lots of enzymes in your body that are responsible for doing these kinds of reactions. Some of them act in parallel. So catalase is an enzyme you make in many of your cells, and it can do the same thing. It can break down alcohol into acetaldehyde. And then you also have a series of enzymes that act in groups, right? So we had this first step. Well, maybe we have a second step in this process. In this case, we turn acetaldehyde into acetate using a different enzyme, a new Pac-Man who comes up. This can easily be converted into fatty acids, and that's why alcohol makes you fat, because you just go 2 little quick steps and you can turn it into something that can be stored for long-term energy in your bodies. So we've sort of talked about these enzymes and I've gone through this lecture. So at this point, I want to see if you guys are with me. So I ask a top hat question, which asks about acetaldehyde. Now, I've made it pink in this picture for a reason, and that is that the symptoms that we associate with a hangover are probably the result of acetaldehyde. It's nasty, toxic chemical, and that's why your body's spending a lot of time making this stuff so it can digest it. If it builds up in your body, it causes face flushing, sweating, heart racing. It's a really unpleasant chemical, and so that's why your body is spending all this time doing it. So it turns out that individuals who are suffering from alcoholism are often given a compound that prevents that enzyme from functioning, and it's called antabuse, because what happens is that if you take antabuse, your body cannot use that second enzyme in the process. It is prevented from working. So at this point, this is the kind of question I ask students to see, what would that look like? You saw what alcohol looked like over time. So now put yourselves in the place of someone who's taking antabuse, and I'll make this very large. And this is now, instead of alcohol, it's showing percent acetaldehyde and time in hours. So what do you think it would look like? So I usually let students vote once, and then I'll say, turn to your neighbor, explain to them what answer you chose and why you selected that answer. Okay. All right. So at this point, I'm usually gratified because you guys are actually talking about this, right? How exciting is that you would actually talk about it? And then I know there are tons of answers out there because I can look and see what people have put different answers. And so I'm actually really curious, right? This is a real issue. Students say, oh yeah, I know about the graphs. I understand how to read them. But then when I ask this question, suddenly they're like, oh, I don't understand. Because which enzyme are we talking about and what's happening? And very often I will ask for groups that, someone who chose A, explain to me what you're seeing in A. And they'll say, oh, well, it started working, but then the antabuse, and I don't know what's happening. I'm sort of confused. I don't really think I understand what's happening. So we'll then select another group, right? You guys had some great comments. Tell us what you were thinking about that.
Well, we were talking about the fact that, we chose C and said that in this case the acetaldehyde would build up. And based on what was being said earlier in the lecture, we were worrying about the links between acetaldehyde and whether it was cancer causing.
Yeah, exactly.
So, therefore, was taking antabuse potentially cancer.
So and I would press them and say, well, how did you know, when you say it was working, you think, oh, well, what does it look like on the graph? So I would have them say, I sort of saw that this level went up and then it never disappeared. Is that what's going on? So that they would understand that. And then I'll ask anyone if they have any confusions or misunderstandings about it. But before, when I had given these lectures and I used to just talk about it, do you guys want to see how you did? Let's see. I'll just show you. Oh, very good. So I can look at this as an instructor and see that there are some students who are still confused. In my class, my students change their answer. Even though they know that I'm giving them points for answering any question, they still don't like to be wrong. They're like, no, see. They're changing it. So if I flash this up, it'll immediately change. So you guys are way better than them, right? You're willing to admit, like, I was confused about some things. I don't think I understood exactly what she was talking about. So that is helpful. I think that there's a kind of combination of things going on. It's something that's relevant to them. They are suckered in, right? They're all interested in alcohol. And they want to know how it works and what it's doing in the body. So they're very sort of suckered in that way. And then this idea of the antabuse and that, people actually taking this and it's sort of preventing or causing them to have issues with alcohol later is also very intriguing to them. So I ask them that top hat question. And then I move on to the next section, which is I say, okay, if you had done a Google search, you would have come up with several different scientific articles that looked at the genetics of these enzyme variations that people have. And so this one was done by a group in 1991, and they decided to look at Chinese men, and they selected half alcoholic and half non-alcoholic. And then they looked at 3 genes, some of which were described in that blog piece. You may recognize them. This alcohol dehydrogenase is the gene that makes that enzyme. This one as well. These two together, 2 and 3, make 1 protein. And then this one is aldehyde, which makes that second enzyme in the step. And they looked for differences in the types of these genes that these Chinese men had based on whether they were alcoholic or a non-alcoholic group. And so I'll give them a minute and they can talk a bit about, why did they choose this population? Why did they choose Chinese men? Ideas? Why not just regular old guys?
So in the comments before, you mentioned that there's a belief that Chinese
That's right.
man metabolizes ADH faster.
Yeah, exactly. So there's this idea that in this genetic background, there may be some higher frequency of different alleles that cause a problem with how they metabolize alcohol. So that's why they chose this population. And then we talk a little bit about what a genotype is, the combination of letters that they have. And I use this found it on the internet for describing this. It's probably from one of these blogs. It's really terrible, and those of you who are biologists, just try to let go of the fact that the way they're sort of depicting this. But here we have a chromosome, and on the chromosome we have a gene called ALDH. And this is the gene, the piece of DNA that would help you code for the enzyme that would help you digest that nasty old aldehyde that's building up in your body. And what they're saying is you can have different variations of it. You can have this version 1, it's got a little asterisk and a 1. And this person is not developing a flushed face, but they have the little bubbles here, so they've clearly had some alcohol. This individual is kind of flushing, and this individual is really flushing. And so if you look, what they're saying is there's a different allele here. You have the same gene, it's on that chromosome, but a slightly different version of it. They're calling it asterisk 2. And so I describe this to them, and I say that's really what they're looking at. This is the genotype, is whether you have 2 of the pinks, a pink and a yellow, or 2 yellows in that sense. And then I show them a table of data from the paper. And in this paper, it has the genotypic frequency. So it has if you went through and counted up, how many did you have that had 2, like this, of your Chinese men that you looked at? How many had 2 of the yellow, version 1? How many had 1 of each? And how many had 1 of each of the pink versions that causes you to flush? And then I further describe that they put these as numbers, and then the numbers are pretty easy to understand. In each case, they had 50 men, 50 non-alcoholics and 50 alcoholics. And so they looked within these men and they said, how many of them had 2 of the number 1s? That was 26. So 26 divided by 50 is 52%. So they just wrote it as 0.52 in the graph. And so you can see they went along for these non-alcoholics and they genotyped all of them and came up with these different frequencies within this population. So this is the data that they would get if they looked at this paper. I have them look at it, and then I say, what are you concluding from this? And I have them do it on a table, usually give them this table, and then I say, here, is 1 more frequent in alcoholics or non-alcoholics? So you guys can go back, and what do you think? Is 1 more frequent in the alcoholics versus the non-alcoholics, and which one would that be? So go ahead, take a minute, talk about it with your newfound neighbor. All right, I'll pull you guys back. So what do we think? What kind of trends are you noticing? I would have them click in, but I'll ask you guys to just tell me what trends, which one do you think? If you had to go to this table, which one, version 1 or version 2, if I had to say which one's more common in which group, what would I say?
1 in alcoholics.
You think 1 in the alcoholics, so we'll go back. Yeah, look, 88 versus 52, and it has a little d there, which means that what they're saying is statistically, that difference is significant. That it's more likely that this allele combination would be found in alcoholics than non-alcoholics. But my students are like, that? They're making conclusions based on that, right? 50, it's not, they want it to be 100%, right? They want it to be cut and dry. Get a genetic test, they tell me. So later in the semester, we have them go to 23andMe, the company that genotype analysis for people, right? You could send your DNA in. And I say, this is data they're using, right? They're just telling you have a higher probability that if you had this particular genetic combination that you would be told by them you have a 1.2 times instead of 1 time chance of having alcoholism. So we talk a little bit about the inconsistencies that you see, the problems with data in science. And then we get back to this idea of, okay, so let's say number 1 is the version that's found in alcoholics. What do we think about the glowing or not glowing in those people? So someone who develops alcoholism, do we think that they're likely to also have that glow phenotype? What do you think? No, it's unpleasant, it's horrible. It makes you feel awful. I have many friends who have it and it's not very nice at all. So we would think that they would not be glowers. And then we move on, we say, okay, this group, non-alcoholics, well maybe they would be the individuals that did have the flushing reaction. So then we start to put it all together. We say, okay, if the production of acetaldehyde is what's causing that, what does this enzyme do? ALDH, what does it do? If you can remember back. Yeah, it breaks this down, right? So then I have them go through with their group in their table and fill out, what does that mean? Does that mean this enzyme's working well in someone? Someone who has the flushing, the red flushing. Is this enzyme working well or is it working poorly? Poorly. So that's their hypothesis. And then the next paper that I have them look at is what do we know about the velocity of that enzyme and how it works? And we also do the same thing for the ADHs. What would happen if this enzyme was working poorly? This guy isn't working. Yeah, you got more alcohol around. And you have less of the unpleasant acetaldehyde. So then they start thinking, okay, wait a minute. Okay, so that enzyme was working faster. The version of that might be more present in non-alcoholics if it was working faster in that case. So they kind of go through and they have to write me an understanding back to this Lord of the Drinks. Well, what would it mean if you actually had a genetic test and someone said that your enzyme had a certain version? What do I think about that? What would it mean in terms of alcoholism and what that enzyme does? And so back to my sort of original, what I wanted them to understand was, what does it mean if the enzyme works at a faster or slower velocity? Can you explain to me what data you'd like to see that would convince you that somebody was more likely, if they had a particular variant of a gene that coded for an enzyme, whether or not they would do it. So to me, I've gotten in full circle back to where it was that I wanted my students to be, but I got them through it by sort of intriguing them and having them work through it on their own without just, there's some telling, but not without telling them the entire thing. Instead, I wanted them to grapple through. Do all of them get it in those classes? No. But they come to me after with their sheet and they say, I really need to understand this. Can I talk to you about this? I'm not sure I understand what it means if this gene is working faster. I'm so confused. And so I can sit down and talk to them about that. As opposed to a normal class where I went through and described what enzymes did and then asked them similar questions, they wouldn't have come to the realization that they had some gaps in where their understanding was. So I gave you that as an example. For you guys, I wanted on your green index cards for you to sort of reflect to me, now that you've had this experience, what kinds of benefits might it have, and then also what kind of problems might it have for you as an instructor. So these are for you to reflect on what just happened and how you experienced it. So these cards are one easy thing that you can do cheaply. Sometimes we'll say students turn them in after class, the muddiest point, for example, or some other challenge that they have. So it'll give me as an instructor an idea of sort of where people were and what they were thinking. It's totally anonymous, your name's not on it, and so I can just sort of look through them at the end and see where students were and what kinds of challenges they had. Judging, and obviously we have more time, but I would say my suggestions would be to start with an important concept that students struggle with. An easy, really easy thing is a test question that they just can't seem to get right. And think about how you might take that test question and turn it into an activity that students could use in class. And how can you get them to do some of the work that you had been doing so that you can tell when they're going south in that. So the example, this is sort of a decision tree that sort of encompasses a lot of what I was talking about today. So you're going to look at the learning outcome for whatever that material is. And you're going to ask yourself, can students achieve this outcome on their own? If the answer is yes, right, then you say, okay, if they can achieve it on their own, maybe this is something that's great for them to do outside of class as reading, or with some guiding questions or some even practice questions that I could give them so that I could sort of help them. You might want to ask, is it going to be necessary to help support that? If it is necessary to help support that, you can make some activity that goes with it that they might be able to view online. If you feel like students are not able to do this, I've given them this test question, they cannot seem to get it, then you need to think, okay, should they be able to figure it out for themselves? Is it an important part of their learning to kind of go through some of the mistakes? And if the answer to that is yes, then just turn it into a question or a problem as part of an activity. If it's not really important for them to figure it out on their own, if they really need somebody to just show them how to derive the equation, that's where your job is, right? Then you can go in and show them that portion of it. But always giving them an opportunity to do some practice in other areas, things that they can learn on their own. So I'll use this is sort of a final example. This is a colleague of mine, Randy Phyllis, who works at the University of Massachusetts at Amherst. And he was teaching an advanced cellular biology class. And in cell biology, in this case, this is a question about the separation of chromosomes during cell division. And it's a big issue because how do you make sure that all the cells, once they divide, they get the right number of chromosomes and they get one of each of the chromosomes, not some sort of combination. And so he was trying to sort of describe to them the various proteins that are involved in this process. And he was also trying to get them to understand that it's a cascade, that there's kind of checkpoints in the cell that help check to see that things are done. So for example, here, the checkpoint is whether or not this center of the chromosome gets attached to the spindle that goes to the opposite end of the cell, so that one would go to the right cell and the other would go to the left cell. And he's sort of describing to them how would the cell know when things had been attached. And then he went through and described several different proteins that are involved in this process in a cascade. It's super complicated. That's just the way cells are. They're very complicated. There's lots of things going on. And no matter how many times he went through it, he couldn't seem to get students to understand and appreciate that. So what he did was he said, okay, I'm going to go through, I'm going to lecture to them on what this is. But then I'm going to simply ask them at the end in pairs to go through a series of true-false questions. What would happen if, for example, there was a mutation that interfered with this protein here ability to bind to happy face? And so the students then had to sit and grapple with this complicated arrangement of proteins and what they do. All the information was there. He had what they do and how they do it. All I wanted them to do was be able to sort of put all the pieces together. So together they had to come up with, well, would this securin protein have this thing put on it? Would this guy have this put onto him? Would this guy stay bound over here? These kinds of questions. So it sort of taught them the content, but then they had to answer really simple true-false questions related to it, and he could sort of have them go through that in his class. So for me, I'm kind of curious, use that kind of decision tree, take a topic that you've been doing passively, but that you think is really important, and maybe write for me what you're thinking in terms of ideas that you have about modifying it. And if you can just write me a very brief one, but for yourself, you might take a chance to sort of make some notes of stuff that you're thinking right now. It would be kind of a cool way to do this in your own classes. All right. Okay, so does it require any class time? Are you doing something that students should be doing? These are all sorts of things that are generally really great questions to start with if you're thinking about doing this as an activity. And then I wanted to end with this. It's some data from Bill Wood, who was in the video that was talking about active learning, started doing changes to his class in 2003. And you can see in this graph, in the fall of 2003, he was doing a very traditional class. And he looked at where students were with normalized learning gains for this course. And they were, it was a bell curve. But he thought, maybe I can add some more interactivity and see if I can improve things. And so in the spring of 2004 is this orange one. He started adding some interactivity, and he saw some gains. But then in the spring of 2005, he added a little more interactivity. He perfected it. That is to say, it's not easy to do overnight. Small incremental changes can have a much greater effect over a long period of time if you kind of keep at it and keep thinking about what is working. It was nice because they had the kind of really good test questions that would tell them where students were doing better and where they still were struggling, so that can be a help as well. And many disciplines have better of those kinds of tests than others. So it sort of depends on where you're at. And I will end there so we can have a few minutes of a coffee break and take any questions.